Hello students, welcome back to your botany class once again, and probably this will be the last topic for this chapter. With that, some topics have been deleted, so this will be the last topic for this chapter, and of course the section A of unit 2. Okay, regulation of gene expression. We have discussed that DNA contains the genetic information which codes for the proteins of all, which codes for all the proteins of a cell. However, these proteins which are coded by the DNA may not be required by the cell at the same time. Some genes, you know that genes are segments of DNA. Some genes may be expressed or transcribed at a certain time, and as such, they are not transcribed when they are not required. The product is not required. Okay, so genes expression or transcription may be different at different genes may undergo different rates of expression or at different times depending upon the cellular requirements. Because the cell is not flooded with unnecessary enzymes or unnecessary proteins. Okay, so this means that as in when the cell requires a protein, the gene which codes for that particular protein will be switched on, would be expressed, and protein will be synthesized. So when the product is not required, that particular gene will be switched off. Okay, so it means that the flow of information from DNA to mRNA to protein is regulated it's regulated under a mechanism of switch on and switch off. It's regulated in such a way that when the, as I've said, when the protein when the product of a particular gene is required, that gene will be switched on so that synthesis of that protein or synthesis of the product of that particular gene will take place, and when the demand or when the purpose is fulfilled, that gene will be switched off again. Okay, so there are certain substances which cause the genes to be switched on. There are certain substances which cause the gene to be switched on so that expression takes place. Those substances are known as inducers, and the switching on of the gene is known as induction, and when the purpose or the demand is fulfilled, the gene will be switched off, which is known as repressor. So the level of gene regulation or regulation of gene expression can take place at different levels. It can take place at the level of transcription when mRNA is being synthesized from DNA, or it can take place at the post-transcriptional level when the mRNA transcript is undergoing processing to make it active, to make it mature, and to make it more stable. Gene regulation can take place even at that level, or for that matter, from transport of mRNA from the nucleus into the cytoplasm. And it also can take place at the translational level or post-translational level. So regulation of gene expression can take place at different levels. And Jacob and Monod in 1961, while studying the metabolism of lactose in E. coli, they proposed the operant concept, so which is also known as the leg operant. Okay, and this leg operant is an example of an inducible system where the presence of inducer results in the switching out of the genes while when the demand is fulfilled, the genes are switched off again. Okay, so according to the hypothesis of Jacob and Monod of the operant concept, they suggest that any metabolic reaction taking place in prokaryotic cells, is controlled in a coordinated manner by a set of genes and not a single gene. Any particular reaction or any metabolic reaction taking place in a prokaryotic cell is regulated or is controlled in a coordinated manner by a set of genes and not by a single gene. So this set of genes which coordinately function to serve the control of a particular metabolic reaction is known as operant. Thus, operant is a segment of DNA which consists of a set of genes or some genes or few genes which coordinately function and serve to control a metabolic reaction. So in this case, they proposed their concept studying the metabolism of lactose in E. coli. And according to them, the leg operant or lactose operant, according to them, consists of the following: the structural genes, the controller genes, and other components. The leg operand consists of structural genes, controller genes, and other components. And these structural genes are denoted by the letters Z, Y, and A. If you have your textbook, you can open up and follow me with that. The letters Z, Y, and A, and these structural genes are arranged in a linear sequence, and they are the regions which codes for particular amino acid. They code for particular amino acid, which will be transcribed, translated into proteins, and which will be modified to form enzymes. Okay, and it consists of controller genes, and the controller genes consist of the operator gene, then the promoter gene, and regulator gene. The operator gene, promoter gene, and regulator or I gene, they are arranged in linear sequence, and this operator gene falls in between, or it comes in between the promoter region and the first structural gene. And of course, these structural genes, as I've said, they code for amino acids which would be transcribed, translated into proteins, and the structural gene Z codes for beta-galactosidase, Y code for the enzyme permase, and the structural gene A codes for trans-acetylase. And the enzyme beta-galactosidase is responsible for catalyzing the hydrolysis of lactose into glucose and galactose, because we are concerned with the metabolism of lactose in E. coli. Okay, so now this operator, it's a short segment of DNA which consists about 27 base pairs, and it's arranged in between the promoter and the first structural gene. And this promoter gene is the site where the RNA polymerase, where it's the site, as we have been discussing, the promoter site is the region, or it's the site where RNA polymerase go and binds, and then it starts its voyage there and initiate the process of transcription of these structural genes. Then this regulator gene, which is denoted by I or R gene, regulates the activity of this operator gene. It has it consists of a sequence of amino acids, or it codes for a particular protein molecule which is known as repressor. This repressor regulator gene codes for, or it synthesizes a protein molecule known as the repressor, and this repressor molecule has a high affinity for this operator gene. It has a high affinity for the operator gene. Okay, so this is the picture of, or the structure of the leg operand. And the other components are the repressor molecule, as I've said. It's a protein molecule which is coded by the R gene or the I gene, and inducer. Inducers are small molecules, the presence of which can trigger or which can lead to the switching on of the genes, or the absence of which can determine whether the genes to be switched on or switched off, whether the genes to be ascribed, to be expressed, or to be switched off. Okay, so now in this leg operand, in the absence of inducer, or in the absence of substances which causes the switch on or switch off mechanism, in the absence of inducer, this repressor molecule, which is coded by the I gene or the R gene, as I've said, they have high affinity for the operator gene. They bind to the operator gene and prevents RNA polymerase from synthesizing or from transcribing the structural genes. In the absence of inducer, when lactose is absent in the medium, inducer in this case is lactose. So when lactose is absent in the medium, this repressor, which is called by the R gene or the I gene, it binds to the operator gene and it prevents the RNA polymerase from transcribing the structural gene. RNA polymerase, which starts its voyage from this promoter region, it could not cross over to transcribe this, to transcribe the structural genes, and hence there is no synthesis of the proteins or the enzymes. Okay, in the next step, in the next experiment, in the presence of inducer, that means when lactose is added into the medium, when inducers are added into the medium, these inducers, they go and bind to the repressor molecule. Repressor which have been transcribed from the I gene or R gene, the inducer go and bind to the repressor molecule, bringing about a conformational change in the structure of the repressor molecule. So the binding of the inducer and the repressor molecule brings a change in the structure of the repressor. When there is a change in the structure of the repressor, it is unable to go and bind to the operator gene. It is not unable to go and bind because there has been a change in the structure. Okay, so now this inducer binding to the repressor molecule makes it inactive, and as such, the inactive repressor will not bind to the operator region. So now this operator region is free. It's free. There is no any obstacle for the RNA polymerase to go crossover. So now this RNA polymerase, which is responsible for transcription of these structural genes, they start its voyage here in the promoter region, they bind to the promoter region, and they go and transcribe the structural genes. They start transcribing the structural genes, and transcription of this structural gene takes place forming leg mRNA, lactose mRNA. Then after transcription, if required, in case of eukaryotes, it's required transcription modification. But in case of prokaryotes, the mRNA transcript does not require to undergo any processing because they are stable, they are matured, they can immediately undergo translation. So transcription of this structural gene takes place catalyzed by the enzyme RNA polymerase, and as such, this enzyme, this gene Z gene codes for beta galactosidase, the Y gene codes for the enzyme permease, and A gene codes for trans-acetylase. So these enzymes are synthesized, and this, as I've said, these enzymes causes the beta galactosidase causes the hydrolysis of lactose into glucose and galactose. So this is how the bacteria E. coli, they are able to metabolize lactose. So the presence of lactose, which acts as inducers, they are responsible for bringing about of switching off of the structural genes. The presence of this causes the RNA polymerase to go and bind to the promoter region and bring about a transcription. So they act as inducer, or this is an example of inducible system. And when lactose is absent in the medium, as I've said, the repressor molecule, which has a high affinity for the operator region, they go and bind to the operator region, and as such, RNA polymerase cannot go and transcribe the structural genes. So the transcription of the structural gene is switched off. So this is how the expression of the genes are regulated. The expression of the genes are regulated in a switch on and switch off mechanism. So the other example of this is an example of a inducible system, and tricoperon or tryptophan operon, the metabolism of tryptophan, is an example of repressible operon. Okay, so this is the last topic of your unit, of your chapter, this. And in the end, I would just like to give a brief review of all the chapters, all the topics that we have discussed from this chapter. We have discussed about the search of DNA as a genetic material. It all began with this Griffith. He started the experiment using those the pneumococcus bacteria in the mice, and how that is able to transform, and how it proved to a certain extent that there is a transforming principle. Then Hershey and Chase, they concluded the experiment through their using this bacteriophage T2, that they concluded that the genetic material is actually not RNA or protein, but it is the DNA. And in case of prokaryotes, the DNA is present in the region of nucleoid, in the nucleoid region, while in case of eukaryotes, it's present in the nucleus because it has been bound by a nuclear membrane, giving rise to a true nucleus. Then next is that we have discussed about this protein synthesis, where the expression of genes, you know that DNA contains the genetic information, and all the phenotypic characters of an individual are expressed in the form of proteins, which are determined by the sequence of amino acids in the DNA. So this formation of proteins to determine our phenotypic characters, the synthesis of which takes place in two steps, transcription and translation. And this flow of genetic information from DNA to mRNA is known as transcription, and from mRNA to protein is known as translation. So in case of prokaryotes, transcription, that is a formation of mRNA from DNA, takes place in the cytoplasm of the cell only, while in case of eukaryotes, it takes place in the nucleus. And then after that comes the process of we have discussed about the genetic code, how the different codons codes for the different amino acids. We know that the codons are a sequence of 3 nucleotide base pairs, and how these 3 nucleotide base pairs codes for a particular amino acid. And there are 20 different types of amino acids coded by at least 64 codons. We have discussed about that. So at least there are 3 stop codons and 3 or nonsense codon which do not code for any amino acid, and there is one particular initiating codon, the AUG. We'll discuss about that. And then we also have discussed about the different types of RNA. We discussed about the mRNA, which is being transcribed from DNA. Of course, all the different types of RNA, they are transcribed from DNA, and the undergoes modification, resulting in the formation of the 3 types of RNA. So we have discussed about mRNA, messenger RNA. We discussed about the transport RNA, and then we also discussed about the ribosomal RNA. Together with ribosomes, together with proteins, they form the structural components of ribosomes, which are the sites of protein synthesis. Then tRNA, which is responsible for bringing the amino acid from the cytoplasm into the ribosomes, which are the sites of protein synthesis. We discussed about that, and we also have discussed about the second phase of protein synthesis, that is translation. How the language of nucleotide bases in mRNA is translated into proteins, into the language of proteins through the process of activation of amino acids, then attachment of that activated amino acids to tRNA, then formation of the polypeptide chain and chain elongation taking place through initiation, then peptide 1 formation, termination, and how that newly synthesized protein undergoes modification to become active. They undergo modification even in the Golgi complex or in the even in the endoplasmic reticulum. Okay, so once it's matured, one is active, the proteins, they are released into the cytoplasm in case of free ribosomes, and when they are attached with the endoplasmic reticulum, they are released into the lumen, and they undergo further modification to assume different parts, or some of them even forms enzymes. And then finally is how the genes are expressed. How that genes are segments of DNA, and that a particular cell requires the protein at different times, require different types of protein at different times, and how the cells function in a very coordinated manner to genes, the genes function in a very coordinated manner to synthesize a particular kind of protein which is demanded by the cell. And when the cell is not demanding that particular protein, the gene is switched off. So the cell, which our human body is made up of, it's made up of trillions of cells. So they function in a very coordinated manner. All the cells are similar. Okay, all the cells have the same genetic constitution, have the same sequence of bases, except the sex cells. So how they function in a very systematic manner that synthesize specific kind of protein only in specific area. So this is how the process of genetic search begin with the formation of proteins, thereby regulating all our phenotypic characters and resulting into our phenotypic behavior. So with this, I conclude this unit, that your unit molecular basis of inheritance, part A. That is part A. Part B, I leave it for zoology. So with this, I conclude this chapter, molecular basis of inheritance. Thank you.